Assalamu alaikum warahmatullahi wabarakatuh. We are studying the biochemistry, and here today's lecture is about the metabolism of amino acids. First of all, we will discuss the transamination reaction, oxidative deamination reaction, and the production of ammonia, its transportation, and its urea cycle. Three different forms of nitrogen are there by which they are excreted, that is the uricotelic, that is present in mammals. Then we have uricotelic, that is present in birds and reptiles. Here uric acid will be formed, and ammonotelic, which is present in the fish, and ammonia is formed. This is the summary of amino acid degradation, and here you can see that amino acid is degraded in two components. One is carboxylic group and another is the amino group. And basically this is an alpha amino acid. This alpha amino acid is get converted into alpha keto acid, and this alpha keto acid is basically undergo another pathway, which is the ornithine citridine cycle. Arginosuccinate is an intermediate compound that exists in both cycles, that is urea cycle and the citric acid cycle. Here arginosuccinate, when undergoes citric acid cycle, it will convert into fumarate, then malate, then oxaloacetate, and then transfer of keto group, and again aspartate will be formed. But on the other hand, the arginosuccinate will be produced from citrulline, and this arginosuccinate will convert into arginine, and this arginine will liberate urea and convert into ornithine, and this ornithine is converted into citrulline, and then again carbon dioxide will be incorporated and ammonia will be incorporated, and again citrulline will be formed. This is the brief idea कि normally ये दो cycles simultaneously चल रही होती हैं. What are the major reactions? The major reaction is the transamination reaction, and this is the first step of the amino acid degradation. Here the amino acid transfer from many amino acids to limited number of keto acid receptors. For example, pyruvate hai, alanine hai, all they are get converted towards the alpha ketobutyrate, and the transamination reaction tend to channelize the amino group onto the glutamate. This is the amino acid having carboxylic group and amino group, and aminotransferase will produce keto acid, and this will all occur in the hepatocytes. It's a reversible reaction in which alpha amino group of one amino acid is transferred to the alpha keto acid already present in the body, resulting in the formation of new amino acids and new keto acid. It is the general process. The amino acid is transferred from this carbon compound or carbon of one donor towards the alpha keto acid, a recipient, forming a new keto acid and new amino acid. Donor amino acid becomes a new keto acid after losing the alpha amino group, while the recipient keto acid becomes a new amino acid after receiving the amino group. The process represents only an intermolecular transfer of ammonia group without the splitting out of the ammonia. Actually देखा जाए तो ये भी deamination ही है. Transamination में एक कंपाउंड से पूरा अमोनिया निकल रहा है, लेकिन ओवरऑल रिएक्शन के अंदर चूंकि अमोनिया बाहर नहीं आ रहा, अलग से सेपरेट नहीं हो रहा, बल्कि ट्रांसफर हो रहा है वन फ्रॉम वन कंपाउंड टूवर्ड्स द अनदर, so that's why it is called as the transamination reaction, not the deamination reaction. The enzyme is alanine transferase, alanine pyruvate transaminase, or glutamate transaminase, depending upon the enzyme, and coenzyme is the pyridoxal phosphate. The next step is the deamination, in which in this process the nitrogen of amino acid is completely removed and ammonia will be liberated. It is of two types, oxidative deamination and Non-oxidative deamination. In oxidative deamination, the site of oxidative deamination is studied. Here, the amino acid is oxidatively liberate ammonia from these amino acids, and furthermore, it also depends upon the nature of the amino acids. It is occur in various organ of the body like kidney, liver, and active tissues. Mostly two enzymes are present. This, these enzymes are amino acid oxidases. They are auto-oxidizable flavor proteins, means जो oxidation है वो automatic है, depends upon the presence of amino acids. And the reduced flavor proteins are reoxidized at a separate level directly by the molecular oxygen, forming hydrogen peroxide without any involvement of the cytochrome of electron transport chain or electron carriers. Immediately खुद ही अपने काम करते हैं. जो हाइड्रोजन peroxide बनती है, it is toxic to cell and it has to be converted immediately towards the water and oxygen by the enzyme catalase that is usually present in the blood. This oxidative deamination is take place in two steps. First, the amino acid is dehydrogenated by flavor protein enzyme, and then in the forming alpha amino acid, and in the next step, the water molecule is added spontaneously and decomposes the corresponding keto acid with the loss of alpha amino nitrogen. Another enzyme is the glutamate dehydrogenase that release ammonia for the urea synthesis. It is one of the few enzyme जो कि NAD और NADP यूज कर सकता है और इसके अंदर हमारे पास वेरियस एंजाइम इनहिबिटर एंड वेरियस एंजाइम एक्टिवेटर भी हैं that either inhibit or activate this enzyme. In deamination, you can see that glutamate in the presence of glutamate dehydrogenase will produce alpha aminoglutarate, and then this alpha aminoglutarate is thus converted into ammonia. Another separate molecule will be there and alpha ketoglutaric acid will be formed. Separate ammonia is there, इसलिए हम इसको कहेंगे this is toxic and this has to be converted by urea. This has to be converted into urea by urea cycle. ये अगर यही यूं ही रहेगा अमोनिया तो फिर ये बॉडी में टॉक्सिक इफेक्ट्स प्रोड्यूस करेगा. This is the oxidative deamination in the presence of enzyme catalase, and here amino acid is converted into first oxidized form and the flavor proteins are oxidase, auto reduce or they auto oxidize the compound and forming alpha amino acid and then alpha keto acid. Here the corresponding enzyme is catalase which converts from hydrogen peroxide into oxygen and water. Second type is the non-oxidative deamination. Here certain amino acids are there which are non-oxidatively deaminated by the specific enzyme and forming ammonia. इसमें भी अमोनिया ही बनेगा, बस जो प्रोसेस है वो ऑक्सीजन के प्रेजेंस में नहीं हो रही, ऑक्सीडेशन नहीं हो रही. Three types of हमारे पास non-oxidative deamination pathway हैं. For example, amino acid dehydrases, deamination of histidine, amino acid desulfhydrases. ये कुछ enzymes हैं जहाँ पे अगर deamination reaction हो रहा है वहाँ पे oxidation नहीं हो रही. amino acid dehydrases, deamination of histidine and amino acid dehydrosulfurase. अगर हम amino acid dehydrases की बात करें, the hydroxy amino acids like serine, furamine, and homoserine, they are deaminated by the specific enzyme called as amino acid dehydrase, which requires pyridoxal vitamin B6 as a coenzyme. ये जो एंजाइम है ये फिर स्पॉन्टेनियसली डीअमाइनेट करता है. सेकेंड हमारे पास हिस्टिडीन की डीअमाइनेशन, that is the non-oxidative deamination by the specific enzyme histidase. and it forms ammonia and urocanic acid. थर्ड है हमारे पास amino acid desulfhydrases. It's deamination of sulfur-containing amino acid like cysteine and homocysteines. Here you can see that L-cysteine is degraded in the presence of vitamin B6, and here hydrogen sulfide is phosphorylated. Intermediate amino acid will be formed, and this amino acid is further converted into another inactive compound and forming the, that is the pyruvic acid, forming the ammonia. In the next example, you can see the in serine. In the presence of vitamin B6, amino acid will be formed. This amino acid will create ammonia and pyruvic acid will be formed. Then we have another chemical reaction which is called as transdeamination. Transdeamination is the deamination of L-glutamic acid. It is noted that L-glutamic acid is not deaminated by the L-amino acid oxidase, but the specific enzyme is there which is called as L-glutamate dehydrogenase. This enzyme has a 4 polypeptide chain, a tetramer, and it is a zinc-containing metalloenzyme. One zinc atom is present in each peptide chain, and it is widely distributed in human tissue. Highly active enzyme, and it is basically specific for L-glutamate. And it also requires NAD or NADP as a coenzymes. What is the reaction? The enzyme L-glutamate dehydrogenase catalyzes the deamination of the L-glutamate to form alpha-aminoglutaric acid, which one addition of a molecule of water forms ammonia and alpha-ketoglutarate. It is to be noted that reaction is reversible and the equilibrium constant favors the glutamate formation, and the quick removal of ammonia to form urea in the urea cycle and alpha-ketoglutarate to a TCA cycle that further favors the onward reactions. This is the reaction here. L-glutamate is there. Here 2 side chain CH2CH3 group is there, 1 amino group is there, and 1 carboxyl group is there in the presence of L-glutamate dehydrogenase. Furthermore, this ammonia group is removed after the addition of water. Here, by the process of water, here carbonyl group will be formed. This transdeamination is a reversible reaction. This is the summary, that is the removal of nitrogen from L-amino acid. This is the alpha amino acid. It first undergo transamination forming alpha keto acid, and on the same side alpha ketoglutarate is converted into L-glutamate in the presence of vitamin B6. This alpha ketoglutarate in the presence of enzyme L-amino acid oxidase converted into keto acid and then liberated ammonia. This L-glutamate is undergo transdeamination forming the L-glutamate dehydrogenase that produces the ammonia. It means कि ammonia हमारे पास बहुत सारे metabolic pathway से generate हो रही है, alpha amino acid से भी generate हो रही है, alpha keto acid की formation के बाद L-glutamate से भी ammonia generate हो रही है, non-oxidative deamination से भी ammonia generate हो रही है, और glutamine की हाइड्रोलिसिस के जरिए भी अमोनिया जनरेट हो रही है. इसका मतलब है बॉडी में फिर अमोनिया प्रोड्यूस होती जा रही है जैसे जैसे अमीनो एसिड का मेटाबॉलिज्म हो रहा है, और ऑल अमोनिया विल अंडरगो दी यूरिया साइकिल फॉर्मिंग दी यूरिया by the addition of carbon dioxide. This is all about the first lecture of the metabolism of amino acids. Thank you.